Good evening. My name is Hannah Antosh, and this is Synaptic Structure and Function for Biology 515, Concepts of Human Anatomy and Physiology 1, for Dr. Sandra Barrera. Before we begin, I want to pose the question, why do we work? Why do our muscles move voluntarily and involuntarily? Why do we feel pain? Why do we feel cold? What is causing us to do these things? Well, the answer is really quite simple. Neurons. Neurons are nerve cells that are found throughout our body within our nervous system. And our body actually contains about 86 billion neurons. And these neurons are constantly communicating. They're telling each other when it's time to move muscles, when we're feeling cold, when we hear something. Our nervous system is constantly working. And they're communicating through things called synapses. A synapse is where two neurons, quote unquote, meet. And I say quote unquote because, as you can see by our picture, there's actually a pretty sizable space between our two neurons. And we'll get to that in a minute. But I want to talk about the blue neuron and the pink neuron. Our blue neuron is what we call a presynaptic neuron, meaning that it is going to be the one that is initiating the signal. Whether it's a chemical signal or an electrical signal, this is where the signal is going to be coming from. And the signal is being transferred through the synaptic cleft into the postsynaptic neuron. And this is going to be receiving the chemical signals, the electrical signals, and our postsynaptic neuron is going to be filled with receptors for specific neurotransmitters. Okay, neurotransmitters are transferred by this synaptic cleft right here. And our neurotransmitters are typically going to be housed in what we call vesicles. And I have vesicles here in this picture. I have them in these gray square boxes. Now, the vesicles typically aren't going to leave the presynaptic neuron. What they're going to do is they're going to leave their vesicle and they're going to travel across the synaptic cleft to the postsynaptic neuron where they will meet and bind with their receptor. For example, GABA A is a neurotransmitter that is an inhibitor. It meets specifically with the GABA A receptor in order to work. Synapses do a lot of things, but what happens when synapses decide not to work? Or rather, when they're forced not to work? I want to bring your attention to Alzheimer's disease. Alzheimer's disease is a progressive neurodegenerative disorder that is characterized by a loss of cognition. And I say these things, my grandmother had Alzheimer's disease, and I always was curious what caused it. Why did she lose cognition? Why did she lose her memories? Well, according to a peer-reviewed article by Wen et al., there's multiple authors, Alzheimer's disease can at least be helped along by something called the complement system. Now, our complement system is made up of plasma proteins that their job is to oppose pathogens, right? Our complement system, we'd like to think of it as the knight in shining armor that's going to destroy invaders. However, the complement system does do things wrong occasionally. And what it will do is it will prune synapses, meaning that it will destroy the synapse through a process called phagocytosis. Phagocytosis is really we can kind of think of it as the cell eating. The cell is going to engulf and like digest whatever happens to engulf. Our immune system does it a lot with pathogens. And in this case, the complement system will do that to these synapses, to these postsynaptic neurons. To the postsynaptic neurons. And when we lose the synapse, this is where we start to see cognitive impairment. Again, this information is all coming from Wen et al. We begin to see this this cognitive impairment due to synaptic loss. Researchers and scientists have identified synaptic loss through synaptic position emission tomography tracers. When doing this, researchers found that the C1q, which is a protein found within the complement system, that information came from Janeway and Murphy from their book Immunobiology, I believe. But when this C1q, when this protein is activated, and we have a lot of it, and we have a lot of C1q, and we have a lot of C3, another protein, they're unregulated with the excitatory postsynaptic neuron PSD35. When we have these two things working together, we can see a lot of times, researchers have seen a lot of times, that this is accompanied by a decrease in postsynaptic density, meaning that they begin to see less of these postsynaptic neurons. Well, that's clearly a problem, right? Because then we have these presynaptic neurons trying desperately to send a message, but no one is there to receive it. That postsynaptic neuron is gone. We also see an increase in phagocytic microglia. Now, microglia in the complement system, their job generally is to clean up after cell waste, and they're going to keep things working the way that they should. Again, this is not an everyday thing. In fact, Alzheimer's disease and synaptic loss can happen to anyone. Again, this information is coming from Wen et al. It can happen to anyone. However, researchers have also found that Alzheimer's disease and synaptic loss is more common in women. Postmenopausal, estrogen production begins to deplete. And as the estrogen production depletes, we see an increase in proteins that lead to the destruction of the synapses through phagocytosis. And again, Alzheimer's disease can be caused by the synaptic loss. It can also be caused by plaque buildup in those synapses as well. It can vary from gender to race to genetic makeup. Alzheimer's disease it's really, really heavily influenced by the loss of synapses. Thank you so much for listening. Here are my sources. I may not have verbally referenced each one of these. I'm very sorry. They should be found down here in this bottom left. This bottom left corner here. Thank you so much for listening, and have a great